So this is my final draft of the science communication project. First of all, what are sourdough starters? Sourdough starters are a mixture of flour and water and maybe some other things. Starters are periodically fed with more water and more flour, and over time the natural yeast and bacteria in the flour ferment together. So again, the lactobacillus and wild yeast ferment together and release CO2, which causes the bread to rise. And lactobacillus gives sourdough that sour taste. And fun fact, lactobacillus is the same bacteria that is found in yogurt, which is why sourdough starters and yogurt can kind of have a similar smell. And without a sourdough starter or another leavening agent like baker's yeast, the bread would just be a clump of cooked flour and wouldn't be light and airy. So in this class, we wanted to investigate the microbial diversity between different fruit starters. Each person had a control starter and one or more fruit starters. And we fed them, we tried to take care of them, and if they survived, we submitted them for genetic sequencing. So for my starters, I chose 2 fruits. Given my luck, I figured that even if I tried to do everything correctly, I would probably do something wrong, and so I thought that if I had 2 fruits, maybe one of them would survive. But inevitably, all 3 of my starters died, and I chose mandarin oranges and Macintosh apples, and my starters started to smell within a few days, and I said before that they died, but in reality, they never grew. They never rose, they never formed hooch, they really didn't do anything except stayed stationary and started to smell. And as you could see, after a few weeks of just sitting, my Macintosh starter here started to grow mold. But eventually, all of my starters grew mold, even the control. So if my classmates had starters that survived, they submitted them for DNA sequencing. But what is DNA sequencing? So the goal is to find out what the order of nucleotides are in the DNA, and this information can then be used to identify different strands of fungi and bacteria that are present in our starters. There are 2 main types of DNA sequencing that we talked about this semester. One was Sanger, which can only sequence 1 DNA fragment at a time. However, it is more cost effective. But the one that we used in this class, because we wanted to find and identify ideally all of the bacteria in our starters, was NGS, which can sequence millions of DNA fragments at one time. So this brings me to my research question, which was, does the acidity level affect the amount and prevalence of microbes in a sourdough starter? So I decided to look at a lemon starter versus a control starter from last semester. I chose this because lemons are acidic, and hopefully I can gain some information about strands of bacteria found in acidic fruits. So I studied the data, and I found that in the lemon starter versus the control, there was a strand of bacteria called Lactobacillus brevis. The sample numbers were 7, which was the control, and 9 was the control number 9 was the lemon. And I found out that L. brevis is found in many fruits, including beer, cheese, and kimchi. So with some research, I found that L. brevis has been used to enhance the taste of sourdough. I found that L. brevis has been associated with citrus and sour fruits before in research. I also found out that L. brevis is avoided in beer and cheese because it can cause beer to go bad, and it can cause unwanted air pockets in cheese. So L. brevis produces acetic acid, lactic acid, CO2, and ethanol from hexose fermentation, and there are some genes that function to survive the harsh conditions of beer. And L. brevis has been modified to produce butanol, which is a proposed fossil fuel alternative from glucose. Given all of that information, I hypothesize that L. brevis will be found in many starters, but especially in lemon and citrus fruit starters. This is because research has associated L. brevis with citrus fruits and because it was so prevalent in the lemon starter. So let's look at the results of this semester's starters. So again, I chose to look at a control versus a lemon starter. The sample number of the control was 112. The sample number of the lemon starter was 111. So this is a graph of the L. brevis that was found in the control starter versus the lemon starter. On the left side, The hot pink bar is L. brevis prevalence. On the left side is the control, on the right side is the lemon. And not only was L. brevis found in both the control and the lemon starter, Lactobacillus brevis ended up having a higher prevalence in the control starter, which really surprised me because I didn't expect that. I expected there, at the very least, to be more of a prevalence in the lemon starter, but it was more prevalent in the control starter. The results do support my hypothesis in a way. I did hypothesize that L. brevis would be found in many starters, and not only was it in the control and the lemon starter, the person's starters that I analyzed also had a raspberry starter, and L. brevis was in that one as well. So I would say that it does support that L. brevis is found in many starters. But I expected it to be even more prevalent in citrus and acidic fruits, which did not happen. So it is hard for me to say whether or not the hypothesis is supported or not supported, and so I do think that there need to be more tests about L. brevis in starters for me to say whether or not I could see my hypothesis being supported. So if this project were to continue, I would want to do experiments that test whether or not Lactobacillus brevis changes to adapt to different environments, and if so, do they affect the starter's ability to survive, and why? But not only that, there are many possible research topics surrounding this. We could talk about mutations, we could talk about the sequencing of L. brevis, we could talk about the effect that Lactobacillus brevis has on the starter. This semester, I believe we were supposed to look at fungi and the fungi data, but we didn't end up doing that. And so maybe the fungi that's present has something to do with the prevalence of L. brevis. Is L. brevis safe? How much of it is safe? How much of a prevalence in the sourdough starter could be considered safe or toxic for humans to consume? Does L. brevis affect the color of sourdough or the sourdough sequencing? Does it have a certain smell? What conditions does it thrive in? There are so many possible research topics just from this one microbe that's found. And I think that hopefully some of this information could be found. Anyways, thank you for watching. I hope you enjoyed this video.